Okay, so in this video, we're going to focus on all things prokaryote. This is for AP biology, but anyone is welcome to watch. All right, so when we talk about prokaryotes, prokaryotes can include both bacteria as well as archaeans, but they are single-celled organisms. But I also want to point out that if you're reading something and it says, or someone's talking to you, whatever, that it's a single-celled organism, it doesn't necessarily mean that it's a prokaryote. You can have eukaryotes that are also single-celled, like a yeast is a single-celled eukaryote, or like an amoeba or something, and so paramecium, etc. So you need to look for other identifying clues, like are there organelles, are there chloroplasts, what are the chromosomes like, to help differentiate whether or not that single-celled organism is a prokaryote or a eukaryote. So in prokaryotes, they are single-celled organisms, and they have a cell membrane, and they also have a cell wall. Now, the cell wall is on the outside of the cell membrane. It is made of it's also made of carbohydrates, but it's not cellulose. So it serves a lot of the same function as a plant cell wall. However, its like building block is a type of carbohydrate called peptoglycan. And you have different classes of bacteria. They could either be gram-positive, gram-negative, and their like cell walls are different, but that's beyond the scope of this class. And then they have one main circular chromosome, where in eukaryotes, we have linear chromosomes. Now, this is not really focusing too much on the genetic aspect, but I should also mention that in eukaryotes, we have introns and exons in our genes, and our DNA is wrapped around histone proteins to help it be organized. That does not exist in prokaryotes. Okay. And then they have ribosomes. So we have eukaryotes have ribosomes, prokaryotes have ribosomes. However, their ribosomes are a slightly different size, but they serve the same function. So when we say prokaryotes do not have organelles, what is more accurate to say is prokaryotes do not have membrane-bound organelles, like a Golgi or a mitochondria or a lysosome. But they do have ribosomes for protein synthesis. Now, a cool thing about prokaryotes is that they have these extra pieces of DNA, little tiny circles of DNA that have anywhere from 50 to 500 genes on them, and these are called plasmids. So these are separate from their one large circular chromosome. Instead, they have in addition, they have these cute little plasmids that we actually, as humans, take advantage of during genetic engineering and biotechnology. So let's go ahead and talk about prokaryotes some more. Now, when we think about back to Unit 4, and we talk about mitosis and the cell cycle in eukaryotes and how our cells divide, that whole process is about dividing the nucleus and making sister chromatids and that all complicated stuff. However, a bacterium has one single circular chromosome, and there's no nucleus to divide. So no, they don't go through mitosis, but they do divide. So how do they divide, right? Well, they need to increase in size as well as replicate their DNA. So here's me showing like they double their DNA. So they also do DNA replication with DNA polymerase, and A goes with T, C goes with G, and the whole leading and lagging strands and all of that. So they will replicate their DNA and increase in size, and then they will go through cytokinesis and they will divide into two daughter cells by a process called binary fission. So when we think about like how would we describe these two daughter cells, well, they're identical, right? We just copied the DNA and divided it in two. So here you have identical daughter cells. And so in the process of binary fission, one bacterium can become 1,000 through exponential growth in just a few hours. Now, if we think about that, though, the bacterium is growing exponentially. And so you would, if there were no mutations or no mistakes, like have a colony of bacteria that are all exactly identical. So when we think about variation and where does variation come from in prokaryotes, there's actually quite a few different processes. So, or processes, I don't know. So when we think beyond just mutations, so mutations, of course, is one of that's like the ultimate source of variation in cells. If we look at us or eukaryotes, we have crossing over, independent assortment, random fertilization. But bacteria have one circular chromosome. They don't do crossing over. So where are some other sources of genetic variation in prokaryotes? Well, there's a process called transformation. There's transduction, conjugation, and transposons. So the next main chunk of this video, we're going to talk about the different sources of genetic variation within prokaryotes. And then at the end of the video, we'll focus on how do prokaryotic cells communicate, right? Okay, so let's go ahead and talk about transformation first. And this was discovered by Frederick Griffith in the 1920s when he was working with pneumonia to try and find a cure. But okay, so in the process of transformation, let's see what happens. Sometimes there can be bacteria in the environment that die, right? And that allows some of their DNA. So in my picture here, the DNA in this bacteria on the left is blue. So as it dies, there might be some naked, it's called naked DNA because it's not surrounded in anything and it's not wrapped around protein like histone proteins or anything. So here you have some DNA in the environment. Now, what a bacterium can do is it can actually take up some of that DNA from its environment and incorporate it within its own chromosome. So now it just acquired new variation. It maybe has new genes that lead to a new phenotype. So here, this bacterium is transformed, like it's changed. It has a new genotype as well as possibly a new phenotype. So this goes hand in hand with those like groundbreaking pioneering experiments on the discovery of DNA, if you are more interested in this subject. Now, sometimes though, those plasmids could also exist in the environment, and we can have the bacterium can actually take in plasmids. Now, this is one of the most common labs that we do in AP Bio, where, not most common, but a common lab to be done in a biology class is called plasmid-based transformation, where scientists can genetically engineer a plasmid so it can they can insert a certain gene or have a gene of interest on that plasmid. And then what we do is we kind of like loosen the bacteria's cell membrane by exposing it to different chemicals and temperatures, increasing the likelihood that the bacteria will take in that plasmid. And then hopefully in the lab you see a new phenotype in your bacteria colonies. So that is plasmid-based transformation, and we'll cover that in a biotechnology video in the future, okay? So the process of transformation is the alteration of a bacterial cell's genotype and possibly phenotype by the uptake of naked foreign DNA from the surrounding environment. Okay, so that's one, or a second, if we count mutations. Now we have transformation as another source of genetic variation in prokaryotes. Now this one is called transduction. So the whole reason I made a video on bacteriophages was so that you can understand transduction. So in transduction, that bacteriophage virus that infects bacteria, when it's replicating during its own lytic cycle within the bacterium, I want to point out here, the bacterial DNA is green and the virus DNA is red. So as these little viruses are reproducing within the bacterium, some of the viruses can accidentally, I don't know why I did quotes, like on accident, incorporate bacterial DNA inside of the virus rather than viral DNA. So now during that lytic cycle, when that virus busts out and goes to infect other bacteria, it's not infecting viral DNA, but rather bacterial DNA. So it's kind of like a little transport from one bacterial cell to another, moving DNA or genes around. So if you did not watch the bacteriophage video I made earlier, I would recommend going back and watching that so you can understand this method of a bacterial cell acquiring new variation. So this bacteria here in the green is going to now move bacterial DNA from one bacterium to another, therefore introducing new genes and possibly like a new phenotype in that recipient bacterial cell. So the process known as transduction, phages or bacteriophage viruses will carry bacterial genes from one host cell to another as a result of errors during the phage reproductive or lytic cycle. That one's probably my favorite. Okay, and then in the next way that a bacterium can acquire new genes or new genetic variation is conjugation. And in conjugation, here we have two bacteria next to each other, and what they can do is they actually can build like a cytoplasmic bridge. So there's actually cytoplasm connecting these two bacterial cells with the cell membrane and stuff. It's not just floating out in the environment. And this is actually called a sex pilus. And so there'll be a donor bacteria. So the one on the left is the donor, and the one on the right is the recipient. And you'll notice, look at that red plasmid. It will actually undergo DNA replication, and a new copy of the plasmid will be replicated and then transferred through that cytoplasmic bridge into the recipient bacteria. And then that bridge is no longer, it like disintegrates or is no longer used. And now that cell has new genes, has new DNA as part of it. So this is sometimes referred to as bacterial sex, but in conjugation, it's the direct transfer of genetic material between two bacterial cells that are temporarily joined. The DNA transfer is one way. One cell donates the DNA and its mate receives the DNA. And then this last topic of transposons, I don't have cute little animations like I normally do because I haven't seen too many questions from College Board about transposons. So it's more of me just like covering all of our bases, but I'm sorry, I haven't put in the time to make it all animated. But anyway, so here you can think of transposons kind of like jumping genes in a way. So you have a plasmid in the bacteria with a section that's a transposon. And what will basically happen is, see this orange area here? It actually can move from one place in the like plasmid into another. Like it can move, like there's enzymes, I believe I have a picture or so. Well, I guess I don't. It can like hop or move and get incorporated into other areas, which would then change the DNA sequence. So transposons do create new variations in genes, and it's basically like a section of DNA moving from one area. I would say the genome, but I'm not even sure if it happens in only plasmids or if it happens in the circular chromosome as well, but a transposon moves from one area to another. I'm sorry, that's an area I am weak on, so if you want to know more about transposons, there's always Google. Sorry. Okay, so now we have learned in Unit 2 about something else that also has plasmids, ribosomes, and divides by binary fission, and that is our mitochondria and chloroplasts. So they both have the double membrane. They both produce ATP. So this is where the theory of endosymbiosis exists, where you have that larger cell engulfed a prokaryote millions of years ago, and now we have the evolution of those the mitochondria and chloroplast from ancient prokaryotes. Okay. Now, when we look at the mechanisms of gene exchange that we talked about, you have transformation, which is the picking up of DNA from the environment. Conjugation is the direct transfer of DNA from one cell to another. And then transduction is when DNA is moved from one bacteria to another by a virus. And that's not on purpose. That's not what the virus wants to do. It just happens by errors in its own cycle. I like this picture because the actual little double helices. I also like this one because you can see in conjugation, you can see how that plasmid is undergoing replication to produce the new DNA strand. And then you have your transformation DNA from the environment getting picked up by the recipient cell. And then you have your phage virus transporting DNA into the recipient cell. Okay, I know I said I was going to talk about cell communication, which is quorum sensing, but I think this needs its own video, so I'll stop here and make a new one on quorum sensing. Okay, good job.